Yes, so I would like to tell a little bit more about using human embryonic stem cells for regenerative medicine, and at the end of my talk I also will go to subtype specification. First of all, an overview of human embryonic stem cells. Well, you've heard about it and you've seen it also in your Hish's talk. So when we talk about human embryonic stem cells, they are derived from inner cell mass from blastocyst stage embryos, which can be then plated on feeder cells and maintained as a cell line. So the characteristic of the human embryonic stem cells is that they can differentiate to all the different, oh, that's not me, all the different lineages. So you can actually differentiate to ectoderm, mesoderm, endoderm, and also to germline, to germ cells. So of course we are interested how we can differentiate these human embryonic stem cells to the cardiac lineage. So if this is just an example of how the embryonic stem cells look like, so this is in a so-called cut and paste method where we have human embryonic stem cell colonies like this, and this is high magnification shown here, and this colony contains thousands of cells. And what you do is after 1 week you can cut up these colonies, and you can then, this is a higher magnification again, transfer them to a new feeder layer as shown here, and this is after 1 day of transfer on a mouse feeder layer. So this is one approach of culturing human embryonic stem cells. Another approach, which is a lot easier, is bulk cultures, which we also have in the lab. So then, of course, how can we make cardiomyocytes from these pluripotent stem cells? So since the beginning, since we have the embryonic stem cells in the lab, we use a so-called co-culture system with endoderm-like cell lines, which is the N2. So it seems to have a life on its own. Anyway, so within this, so after co-culture on these N2 cells, we can actually differentiate these cells within 12 days, and you have beating aggregates like this. So this is what you see after 12 days. And we tried a lot of different things, like different growth factors that you can use to further enhance the differentiation efficiency. And basically it was 2 steps which were very simple to get this efficiency higher, which was leaving out the serum and also leaving out insulin, which gave us a more than 70-fold increase in cardiomyocyte differentiation efficiency. So that was good, and also of course made it possible for us to go to a next step. But first of all, of course, we wanted to see if we have these beating aggregates, how do they look like if we go into dissociation and stain for cardiomyocyte marker. So this is cardiomyocytes derived from the embryonic stem cells, dissociated, stained with alpha-actinin, tropomyosin, and here you can see a higher magnification. You can see the nice striations of these cardiomyocytes, but you can also see that not all of these cells, as shown by the blue nuclei here, that not all of these cells are cardiomyocytes. Furthermore, if you compare it to human fetal cardiomyocytes, you can see there's a lot of resemblance between the two. So, and if you also compare it to the adult human cardiomyocytes, you can clearly see there's a difference. So we already talked about maturation of these cells. So when we make cardiomyocytes from these embryonic stem cells, they more have this fetal-like property. Also, if you look at the action potentials, you can see they have more fetal-like property. So the first of all, so since the derivation of these embryonic stem cells, it was speculated that we could use them as a possible source for cell replacement therapy, and that's also what we've tried in the lab. So the first thing we did was using a human embryonic stem cell line which express ubiquitously GFP, and we've used, after differentiation to cardiomyocyte, 200,000 cells to 1 million cells, and you have to remember that it's not like a pure population of cardiomyocyte, but approximately 20% of these cells, of these beating aggregates, contain cardiomyocytes. So we used immunodeficient mice, so SCID mice. We injected the cells in the heart, and we followed up the expression on to see if there's GFP in after 2 days and up to 13 weeks. We made cryosections, and furthermore we also looked at the confocal microscopy and performed different stainings. So this is a picture of the whole heart after transplantation. So you can nicely see that there was a graft area expressing GFP, and this is a high magnification of that, and no fluorescence showing that's really GFP. We also confirmed that with stainings, etc. So this is then a section of a heart, and you can see that this GFP positive cells, and I hope you can see it, has also striated. So also has the striations of a cardiomyocytes, and this is the overlay. So clearly we could see expression GFP in these hearts, and also striations of these cells. But we also knew that if you look at this particular graft size, that it was not quite enough for to completely regenerate a heart, let's say. But another interesting feature here is when we look at the percentage of cardiomyocytes after transplantation. On the left side, you can see. So remember, we had the beginning at the beginning of the experiment, we have 20% cardiomyocytes, and that's what you see after 2.5 days. If you look for specific markers, you can see that's approximately 20%. But if you look at 3 months after transplantation, you can see that almost all of these cells are cardiomyocytes. The other way around, so if you look at an endoderm specific marker, so this is staining the rest of the cells, you can see at the beginning it's almost 80%, and after time, after 3 months, you can see that almost no endoderm like positive cells are around. So there seems to be selection of cardiomyocyte survival here. So of course the next question is, so if we can have the survival of cardiomyocytes in the heart, so what happens if we use this model of myocardial infarction? So if we inject in this myocardial infarcted heart, can they rescue the heart function? So we performed this experiment. We again we used the SCID mice. The myocardial infarction was created, and we injected GFP positive cells again, so ubiquitously expressed, from either cardiomyocytes or non-cardiomyocytes. So when I talk about non-cardiomyocytes, these are differentiated cells, but they were not beating, and we also checked for markers, and they mostly had these endoderm-like markers. Furthermore, we looked at heart function by MRI, as shown here. You can see the survival of the mice and the MRI images. And I have to say, in the beginning, we were very, we were surprised, but we also relieved to see that after 4 weeks, if you look at the ejection fraction, so the function of the heart, that the decline in ejection fraction was not that severe in the cardiomyocyte group when you compare it to the non-cardiomyocyte group. So that was, of course, very good news. But the disappointing thing is that if you wait for 8 more weeks, so at 12 weeks, we could see that this effect was not there anymore. So we could not see that this improvement was sustained for more than 4 weeks. So this was disappointing, but like I said in the beginning, if you look at the graft side, it was also not surprising that that at the long term cardiac function was not restored. So we decided actually after these experiments that we needed to go one step back, that we needed to understand how we can make these cells, how we can purify cells, and that we know exactly what we have in our culture dish. So we already mentioned, of course, that what you can do with these cardiomyocytes derived from pluripotent stem cells. So if you talk about other possibilities, like you can think of making biological pacemaker. So in that case, you need to know exactly what you have, and that you have pacemaker cells in your culture dish. Of course, you can also use it for other purposes, like using it as an in vitro model for cardiomyocyte differentiation, which we have done, drug toxicity, and also looking at cardiac disease in these cells. But if you look at this picture, and I don't have to explain you that if you go to the different regions of the heart, to pacemaker cells, to ventricular cells, atrial cells, and you look at the action potential, of course you have different cardiomyocytes. So cardiomyocytes have, of course, has different properties. So we believe that if you can achieve homogeneous populations of these cardiac subtypes, that you have a high predictability if you go into the assays or the things that I just mentioned for transplantation, for in vitro models, And if we talk about transplantation, so we already discussed also earlier today that you can talk about homogeneous populations of cardiomyocytes, but maybe you also would like to add other cells like fibroblasts. So of course this is, I'm not saying that we only need homogeneous population, but at least it would be good if we have if we have an idea about the mixture of these cells and what we have in our culture dish. So if we then go back to cardiac differentiation, it's of course what can we learn from embryogenesis. So in this nice review, you can see that during development, when you look at the different markers during these developmental stages, that there are specific markers such as brachyury, MAS1, transcription factors, NKX2.5, islet-1. These are all well-known transcription factors, and if you look at a timeline, it's clear that Wnt signaling is important, and also transient expression of these transcription factors leading to structural markers such as troponin T. So we have also looked in our differentiation system whether we also could pick up these kind of genes. So in this particular experiment, we differentiated human embryonic stem cells, followed it up to 12 days where we have cardiomyocytes, and at different times we looked at microarray expression, and what we could see is that we have clusters of genes which very much resemble the in vivo developmental gene expression. So for example, at day 3 we have genes which are correlated to gastrulation and the formation of mesoderms such as brachyury, MAS1. Day 6 we have cardiac progenitor cells, or genes that are related to cardiac progenitor cells, and at a later stage from 9 days onwards you have genes that are related to the structural genes. So differentiating hESC cardiomyocytes follow a wave of expression which are comparable to the in vivo cardiac development. Another important thing is that that we are able to manipulate these cells. So what we have been doing also in the last couple of years to improve methods for genetic manipulation. So basically, to have a very short introduction to this, is that we change the culture conditions of the human embryonic stem cells and allow them to adapt to these culture conditions. In the final stage you have a situation which you can scale up these cells, and by these culture conditions it's now possible in different lines to efficiently manipulate these cells by different methods. And I have to say now it seems all very logical or normal to achieve this, but a couple of years ago it was not very easy to get high efficiency in all lines. So if we want to go to specific cardiac subtype populations, then of course it would be nice to again look at cardiac development and which genes are responsible for that, and the regulatory elements that are known. So if we look at genes and regulatory elements, well, if we take NKX2.5 as an example, it's very early expressed, but also other genes and regulatory elements can be found, for example, in the heart tube, or specifically in right and left ventricle, or more specifically the left ventricle, but also in regions which are important for the cardiac conduction system. So what we did is we went back to the NKX2.5. So in collaboration with the Monash Institute, we created this NKX2.5 knock-in line as shown here. So this is NKX2.5 locus, and this is the targeting construct where exon 1 was replaced by GFP. So creating this hESC NKX2.5 GFP line. So this line was used for differentiation to cardiomyocytes, and of course what we would expect is in the whole time course of differentiation that since NKX2.5 is upregulated at the cardiac progenitor cell stage, so about here, that we would see GFP expression. And this is the case. So this is an example of a differentiated area in this using this NKX2.5 line, and here you can see the differentiated area that nicely is expressing GFP. And here we actually see that area where these cells were dissociated, and you create a monolayer by doing this. And if you look at this, you can see that a nicely contracted area, but also in between the GFP positive cells you have negative area, so cells that are contracting along with the GFP positive cells. So In the next step, by using this particular line, we also switch to a defined and controlled way of differentiation into a 96-well high throughput manner, shown here. And this is a protocol where more labs are using this. So Gordon Keller, for example, also has this comparable differentiation method. So what we do is, after taking them into differentiation, we use growth factors, and two important growth factors are the BMP and activin A. This is taking place in the first 3 days. Then you have a wash of those growth factors, and at day 7 we plate the cells, and around 2 weeks you have nice contracting areas. And you can also follow the same procedure to create monolayers. So having this GFP line, it's also a very nice tool to further optimize cardiac differentiation. So what you can see here in this particular schedule is concentrations of BMP4, so increasing concentrations, and here increasing concentration of activin A, the two important growth factors for cardiac differentiation. So here at this particular spot, we don't have any growth factors, and you also can see there is no GFP expression. So GFP expression is indicated by the red, by this heat map shown here. So there is an optimal concentration, in this case around 2040 or so, where optimal cardiac differentiation takes place. And I have to say, this is, when you use different lines, this optimum can be at a different point in this heat map. So it's important to improve this for every line. So of course we have GFP, and with GFP we can select for these cardiomyocytes, but another of course what we would like to do is, instead of using a genetic marker, also look for other markers which we can use for purification of these cells. So this is an example of co-staining of, here we have GFP expression co-stained with E-cadherin, and you can see that there's no overlap between these two parts, and this is a bigger, let's see, a whole EB, and you can see that there's E-cadherin expression and here GFP, but it also indicates that there's still areas that are blue which are not expressing GFP but also not E-cadherin. So you can enrich by negative selection in this case, but you also will not have a pure population of cardiomyocytes. So by using this more high throughput differentiation to cardiomyocytes, which gives us more than 50% cardiomyocytes in this format, we also performed, in collaboration with the Monash Institute, microarrays on these differentiated cells, and from this we identified two markers, which is called SERP and VCAM, and we used, and these are cell surface markers, which we use for isolation of cardiomyocytes. So that is shown here. You can see that by having the combination of SERP and VCAM, and SERP is not known, is not related to cardiac development, so it was a surprise to pick it up, but you can use it for sorting of these cells. And here you can see the co-expression with GFP. And also, if you take a look at the different parts of populations of after sorting, so the negative fraction or the SERP and SERP VCAM positive populations, you can see that markers such as NKX 2.5, IREX4, and myosin light chain, etc., are increased in those sorted cells. So in the last part, I want to go to different signaling mechanisms that can play a role in subtype specification. So this is hard development at a glance. We already mentioned some of those signaling pathways throughout development in this morning. So the Wnt pathway, BMP, the nodal, Notch, so these are well-known pathways. And you also see here the transcription factors at the different stages during cardiac development, which have been shown to be important for that time point. So what we try to do is to use again this high throughput method as shown here. And we first of all looked at the cardiac subtype specification, or the distinct profiles, transcriptional profiles in our EBs, and this is shown here. And this is a contracting area, and if you then see, so you can see here the GFP expression again, and if you stain for specific markers like the transcription factor HAND1, TBX5, also in combination with islet 1, you can see that there is overlapping patterns in one case, such as here for HAND1 and TBX5, but not with all the GFP positive cells. And also here you can see that sometimes the cells are not expressed at the same position, so also indicating that you have distinct molecular profiles in our EBs. Furthermore, I want to indicate that it's very important, we already talked about mouse embryonic development, other models, that you also go to human cardiac development and look if these genes that you think are important for cardiac development are actually also expressed there. So this is an example of a tissue section of a 12-week fetal human heart, where we looked at different markers in the sinoatrial node, as shown here, in this upper panel, and the atrioventricular node in the lower panel. So what you can see here is that troponin is expressed in this area. So this is the sinoatrial node, and you can see that NKX2.5, as expected, is not expressed in this area, but HM4, we already heard about this from Kenneth Chen, that is expressed in the cardiac conduction system and also in the sinoatrial node, as shown here, but also still in other areas and also in the atria. So that's why it's important to understand how this is occurring in human embryos. And here the transcription factor TBX3. And you can see the pattern for the atrioventricular node. So with this information, we would like to change our culture system by overexpressing different transcription factors, etc., or change the signaling pathways that are important for heart development. So what has been shown is that retinoic acid plays a very important role in heart development, and this is also something that we have tried in the human embryonic stem cells differentiation protocols. So this is the pathway where vitamin A is leads to retinoic acid, and the main enzyme for that is RLDH2. And it's also known that that retinoic acid is important for restricting the cardiac progenitor cells, as shown here. In an example where you have a RLDH2 knockout, you can see that there's expansion of the heart field. So, and there are multiple studies showing that retinoic acid is important for cardiac development. So if we look at our differentiation protocol and at the different time points, we see that RLDH2, so the important enzyme for this pathway, that is expressed at higher levels at around day 8 and 10. And furthermore, if you look at immunostaining of RLDH2, you can also see that it's expressed in these differentiating cells. So what we did next is to adapt the culture system by adding retinoic acid at the stage where we already have mesoderm, so from day 3 onwards. And we followed the expression patterns of and for example, the beating frequency of these cells after the differentiation. So this is the result of that particular experiment, where we have 2 concentrations of retinoic acid, so 10 nanomolar and 100 nanomolar, and you can see by increasing this dose that you have a higher frequency in this in these differentiated cells in the presence of retinoic acid. Furthermore, if we looked at specific genes, so here is a panel of genes which are more correlated to ventricular genes, we could see that there is actually a downregulation of these genes, such as MLC2V, IRX4, HA2, HAND1. And if you look at atrial genes, we actually see that these genes are upregulated. So sarcoliapin, for example, islet1, HA1 and HAND2, so they are not specific atrial markers, but of course these is an example that genes are related to the ventricular and atrial genes, and there's a switch during this differentiation procedure. So furthermore, if you would like to then go to a functional readout, you, of course, the first thing that you would like to do is look at the electrophysiological profile. But what we did before that is actually look at the ion channels. So here's an example of atrial versus ventricular cardiomyocytes. And if you look at these particular genes here, so this is KV1.5 and KIA 3.1, 3.4, you can see that these are expressed in atrial cardiomyocytes but not in ventricular cardiomyocytes. And when we treat the cells with retinoic acid, we can see that these genes are upregulated in these cardiomyocytes, so confirming that we have a shift towards atrial cells in these cultures. So of course, Then I come to the summary of my presentation. Well, in the first part I showed that human pluripotent stem cells for transplantation, we still need to learn a lot, that it's simply not going to work if we inject cells like this, and that we need to have more defined differentiation cultures, and maybe that we can have mixtures of cells which in the end will lead to improved heart function. I showed you also that we have in this cardiomyocyte differentiation protocol that we have mixture of subpopulations with distinct molecular profiles, that we can use the hESC-CM 2.5 line for homogeneous populations of cardiomyocytes, and then we also can sort it with SERP-VCAM markers, and that retinoic acid leads to an ATRIAL-like phenotype if we do that in the differentiation procedure. And as a last comment, I want to say that combination of these lineage and subtype specific reporters where we're now working on, in combination with fine-tuning of these differentiation protocols, so by adjusting transcription factors or the signaling pathways, we hope to get cardiac subtype populations in the future. So thank you for your attention.